Hello, everyone, and welcome to today's SADS Expert Insights webinar. My name is Guido McGuigan, and I'm the Marketing and Communications Manager of the Canadian SADS Foundation. Thank you for joining us for this important session focused on emerging therapies and research that may shape the future of care for inherited heart rhythm conditions. Our Experts Insights series brings leading clinicians, researchers, and advocates directly to our community to help bridge the gap between scientific discovery and real-world impact for individuals and families affected by SADS conditions. Today, we are pleased to welcome Dr. Gabriel Brooks, Chief Medical Officer at Solid Biosciences, and Seth Rotberg, Senior Manager of Patient Advocacy at Solid Biosciences. They will be presenting on Solid Biosciences gene therapy program for CPVT in the ARTEMIS trial, a first in-human phase 1b clinical study evaluating SGT-501, an investigational gene therapy designed for individuals living with CPVT. Before we begin, I know everyone is already without their microphones and their cameras on, but a few quick housekeeping reminders to please keep your cameras off and microphones muted throughout the presentation. All questions should be typed into the Q&A box. You can submit your questions at any time during the webinar, and we will address as many as possible at the end. And just a friendly reminder that the session is for general educational purposes only and not intended for personal medical advice. So please keep your questions general rather than specific to you or a family member's medical situation. With that, it is my pleasure to welcome Dr. Gabriel Brooks and Seth Rotberg.
Awesome. Thank you so much for the great introduction. I'm just going to share my screen real quick. Let me know if you can see it. Maybe thumbs up.
It looks good, Seth.
Awesome.
Perfect.
Sounds good. Well, thanks again, everyone, for joining us today. And, just an opportunity to learn more about Solid's gene therapy program for CPVT. Just a kind of, I know you already made an introduction, but want to just kind of add a little bit more. So I'm the Senior Manager of Patient Advocacy at Solid, but I've also been a long time patient advocate in the rare disease space after learning about my mom's diagnosis with a rare disease called Huntington's disease. And that's really what's fueled my passion to do the work I'm doing every day, which is connecting with all of you today and trying to learn about how we can further support and advance research. But I'm also joined by my colleague, as mentioned earlier, Dr. Gabriel Brooks, our Chief Medical Officer, but he also leads our clinical function for all of Solid's neuromuscular and cardiac programs. I also wanted to kind of give a huge shout out to the Canadian SADS Foundation for hosting this webinar and for everyone here today for just joining us to listen, learn, and then hopefully we can answer any questions you may have. Before we kind of dive deeper into it, just a reminder that Solid Biosciences is a publicly traded company, and today's discussion will include forward-looking statements that may differ from actual outcomes. So for today's topic, we're going to be sharing a bit more about who we are as a company, how we kind of got started. We'll provide some background on how gene therapy works and why we believe it holds potential for CPVT. And then we'll share our clinical trial overview and kind of dive deeper into some of our preclinical data findings and why we believe this can help further advance our investigational gene therapy, SGT-501. We'll then kind of dive a little bit more into our ARTEMIS study for CPVT, and then finally leave enough time at the end to take questions from the community. So as Guido mentioned earlier, please drop them into the questions or chat box. We are more than happy to answer any questions that you may have. So with that being said, we Solid Biosciences. So we are a life sciences company really advancing precision genetic medicine for neuromuscular and cardiac diseases. The company was actually founded in 2013 by two parents after their son was diagnosed with Duchenne muscular dystrophy at the age of 2 years old. One of those co-founders is the Senior Vice President and Head of Patient Advocacy, Annie Gannot. And really the company was built with and for patients with the mission to deliver meaningful treatments for Duchenne. But since then, we've also expanded our mission to support other conditions such as Friedreich's ataxia and rare cardiac conditions such as CPVT. We work closely with community members and patient advocacy organizations from around the world to advance our mission and really make sure that the patient perspective is included in our everyday work. We're based in Charleston, Massachusetts, which is an area outside of Boston. And our team is united by a single purpose of really bringing life-changing therapies to patients and families who urgently need them. And so with that being said, I have the pleasure of turning it over to my colleague, Dr. Gabriel Brooks.
Seth, thank you so much, and Guido, thank you for having us today to share with you our program for CPVT, which is called SGT 501. To introduce again myself, so I'm Gabe Brooks, the CMO here. I'm a clinically trained cardiologist, and have the privilege of working with a fantastic team to advance therapies for not only CPVT but also neuromuscular diseases such as Friedreich's ataxia and DMD. Our purpose here, we're really privileged to share with you, I think, some basics around gene therapy. What is this prospect of this medicine, these kind of medicines we call gene therapy? If you are well versed here, please do pardon me if I just kind of go through the basics, but I want to make sure that we can all come together and have a point of kind of common understanding. So that's one piece. And then the next is I'd love to share with you our specific approach to catecholaminergic polymorphic ventricular tachycardia that came out of learnings from leading scientists in the field that then allowed for this therapy to come into clinical trials, and hopefully we hope, if safe and effective, that it could provide a therapy for patients. So let's start with talking about what is gene therapy. And first, if we want to talk about what is gene therapy, we really have to start with what are genes. And so genes are, part of our DNA, part of the code that makes all of us. And a gene is really codes for a protein, which is the fundamental building block of course, all of our cells in our body. So DNA is in the nucleus, and so you have to get DNA into the nucleus, where it's transcribed into RNA, which then is translated in the cytosol. So then, if we understand what this thing is called DNA, which makes up the gene, then the next slide is what is gene therapy. So gene therapy is really the process of adding genetic material to an existing cell. And that can be, in one case, a gene that's replacing a gene that's not functioning well, or augmenting a gene that's already there. And that's actually the type of gene therapy that we're talking about with SGT 501, which is actually to supplement the expression of calsequestrin, which we have been able to show leads to an improvement in the function of RYR2, which is the gene that's most commonly mutated in CPVT. So we have this second flavor of augmenting an existing gene by adding more calsequestrin genetic material into existing cells. One of the things that this particular gene therapy does not do is it doesn't alter the genes that already exist in our body. So that's called gene editing. The kind of gene therapy that we're talking about today is not gene editing. And also, this is not a gene therapy that can be a therapy that is passed down to children. This is really something that is added to existing cells and is not integrated into the DNA of the patient. So then, what's the need for gene replacement therapy, and how do you do this gene replacement therapy? So the biggest question, the biggest hurdle is supplying that new genetic material into the existing cell. And what we're leveraging is the power of the virus. So these adeno-associated viruses have developed over our existence on this planet, and it has it has in its shell, what we call the capsid, a way of engaging with cells and being taken up by cells, with then its content being transmitted into the nucleus. And that's how these viruses exist in nature, and that's how they replicate. What we've learned to do is we've learned to keep that shell, that capsid, and take out all of the viral genes and replace it with the genes we want. And so in that way, we've co-opted the virus. And now it's become, if you will, sort of like a delivery truck. It knows where to go. It has the postage on that shell. So it goes to the cells that we're targeting. And then instead of delivering viral DNA into the nucleus, it's delivering the DNA that we've put inside. And that is, in this instance, the calsequestrin gene. Now, with that calsequestrin gene, in this particular instance, there is an on switch, and that's the promoter. So when we want to start transcribing into RNA, which then gets translated into protein, we have a promoter. We have an on switch that turns on in muscle cells. So not only is this capsid, this viral shell, targeting, in this case, the heart, it also has an on switch that only turns on when it's inside of that cardiac muscle cell. And then it is transcribed into this protein, and we call this protein, in this instance, a transgene. So if we go to the next slide of how gene therapy works, this is the visual of kind of what I've walked us through. You have this viral capsid, this viral vector. We call it a vector when it has DNA inside. And in this case, we've taken out the viral DNA. We've put in calsequestrin with a muscle-specific promoter. This virus, as it does in nature, homes to the cell, in this case, the cardiomyocyte. It goes inside, uncoats, and then puts its DNA, but in this case, it's the DNA we've put inside, into the nucleus, where it makes proteins. So in the next slide, let's think about how these kind of drugs work as opposed to traditional drugs we take by mouth. So here, again, the approach is to target the genetic cause of the disease to prevent its symptoms, whereas traditional drugs are used to really manage the biology of that underlying disease. So here we're really trying to target at a genetic level the cause as opposed to the downstream biology. In this case, and in an AAV gene therapy that we know today, it is a one-time therapy that produces long-lasting effects. Whereas, of course, with a normal oral therapy, the drug is only going to work as long as it's in your body, and then, of course, it no longer performs. So how permanent is this? A gene therapy is non-reversible. So when we give a gene therapy, that gene will be in that cell for as long as that cell lives. In heart and cardiac gene therapy, because the cells in our hearts do not turn over, these are cells that are born with, for the most part, of the cells that are going to be there for our entire lives. This really is a therapy that that theoretically should last for the duration of that heart. I will be honest, because I'm sure we'll get a question, but I'll say right now, and I'm happy to talk about it more. How long should you expect this type of therapy to last, especially in the setting of an organ that doesn't turn over like the heart? Well, what we know is these gene therapies do go out many, many years, 5, 10. We're kind of learning as we're going, because this young children that have had spinal muscular atrophy and being treated with a gene therapy for that, they're still showing that that gene therapy is going on. So for as long as we know, our understanding of the length of time that gene therapy lasts continues to increase, because we see the gene therapies that we dosed years and years and years ago continue to be effective. So that is the permanence of gene therapy. What we can say now is that they do last many, many years, and we're continuing to learn, and theoretically that therapy may last for the life of that individual. This is, of course, in distinction with traditional therapy, which of course washes out of our system. When we're testing gene therapies, because it's a long-lasting therapy, the trials are relatively long, and that's because we want to make sure that we can monitor for safety over many, many years, even if we can already make a determination pretty quickly whether or not that gene therapy is effective. So from a safety standpoint, we are committed to following patients for at least 5 years to make sure that we understand the safety of that particular drug. And then, I think when we talk about gene therapy trials, one thing that it's really important to share with anybody that wants to partner with us in the exploration of the safety and efficacy of a particular gene therapy is that by taking a gene therapy, because it's irreversible, that may preclude that particular patient's participation in a subsequent trial. So that is important, as opposed to some traditional drugs. If you're considering being a part of a trial for a traditional oral drug, that may not disqualify that particular patient from future studies. So that, I hope, gives a little bit of an overview of the prospect of gene therapy in relation to more familiar drugs. So what are the potential risks that we think about when we think about putting a gene inside the cell using a viral vector capsid, so with gene therapy? So one of the things we know about gene therapy is that sometimes there can be risks to the liver. And so we use different immunosuppressive medicines to try to decrease the inflammation to minimize that risks. We are giving viruses to people when we think of gene therapy. So the immune system does its job, and it does wake up, and we do see an immune response. Most of the time, patients do feel fluish after gene transfer therapy, but those flu symptoms do go away. When we do give gene therapy to patients, they do produce antibodies, and that's one of the reasons why when I was talking about some of the things you need to think about, what we all need to think about with gene therapy is that it's probably a one-time exposure because you have all these antibodies that are produced when we actually give that gene therapy. So how do we think of mitigating these risks? One thing we do is we do use immunosuppressive agents for a short period of time. We do stop those agents, and so the patient's on their normal medicines after that. We do follow, like in all clinical trials, patients closely in terms of sampling blood and, of course, having them come back into the doctor's office for checkups. And then, as I mentioned, we do keep tabs on our trial participants for at least 5 years just to make sure that everything is going well from a safety standpoint. So that hopefully gives an overview of what gene therapy is and how it's differentiated from other traditional medicines, and then a bit about some of the things we need to think about from a patient standpoint when we think about participating in a trial of a gene therapy. So why are we thinking about gene therapy for CPVT? So I think the first and foremost reason is that we fundamentally have understood in talking with the CPVT patients that better therapies are needed. And I think we're really motivated by the stories of patients that have CPVT in terms of how this diagnosis can be limiting their life and also pose, frankly, a risk of a poor outcome that we, of course, want to prevent. So why now, I think? The reason why we are bringing forth a gene therapy, potential gene therapy for CPVT now, is because the science has advanced in our understandings of what is causative of CPVT and how we might think of changing that biology, of normalizing that biology, has matured. So so that really is, the basis of why we're thinking about a therapy for CPVT, because of the lack of medicines that really affect the root biology of CPVT in the setting of understanding how much continued medical need there is in the CPVT patient. Finally, because we have a better understanding of the biology of CPVT, and we believe that we have new ways of normalizing that biology. So let's talk about, in the next slide, how we are coming to that CPVT abnormal biology and trying to rectify it, trying to make the heart keep in a normal rhythm. So how do we think about CPVT? We really think about CPVT as all about how there is sometimes abnormal calcium handling in the cardiomyocyte, which can lead to unstable heart rhythms. We know that in CPVT, one of the things that happens is during an electrically quiet period of time called diastole, there's an abnormal opening of what's called the RYR2 channel, or the ryanodine receptor, which allows calcium to leak out of the sarcoplasmic reticulum, which is a place that stores calcium in the cells, into the cytoplasm. And what that does is that triggers activity in the cell, and it also makes neighboring cells active. That's so critically important because during that electrically quiet period of diastole, if we do have those abnormally openings in enough cells, that can trigger a new electrical current in the heart. And that is an abnormal and a very dangerous time for that new electrical current to occur. If that occurs, what can happen is we can establish ventricular tachycardia, so very fast and unstable heart rhythms, which could deteriorate, and that could be life-threatening. So we don't want that to happen. We want that heart cell to remain quiet when it should remain quiet, which of course is in diastole. So there are many different causes of that abnormal opening of the ryanodine receptor. The most common is of a mutation in that ryanodine receptor itself, and that's what we call CPVT1. The second most common is a lack of calsequestrin 2. What is calsequestrin 2? Calsequestrin 2 is a protein that hangs out in that sarcoplasmic reticulum, that storage place of the calcium, and it kind of soaks up the calcium like a sponge. And in some patients, they lack calsequestrin 2, and if they lack calsequestrin 2, that means that there's excess calcium that can stimulate the ryanodine receptor to open abnormally. And in both cases, what happens is either if you have a mutation in the ryanodine receptor, that means that it opens more easily in diastole than it should, or we have less calsequestrin. There's still now more calcium hanging around. It's not in that sponge that can stimulate the ryanodine receptor to open abnormally in diastole. So why do we have arrhythmias all the time if we have CPVT? It's because it's really only when we have increased calcium levels that we arrive at a situation where you have that abnormal ryanodine receptor opening. And when do we have these increased calcium levels? Well, anytime our sympathetic nervous system is active. And so that could be under an emotional stimulation of one kind or another, or it could be physical activity. So this is really explains the phenomenon of what we see in CPVT, which is with exercise or with emotion, we have increase in sympathetic or adrenergic impulse that loads calcium into the heart cell, which loads into the sarcoplasmic reticulum, where either there's a ryanodine receptor that opens too easily, or there's not enough calsequestrin around to soak up that calcium. And in either case, that does increase the risk of there being an abnormal opening in diastole, which again, it's not what you want. That can cause that dangerous heart rhythm. So how do we think of actually trying to solve this situation? Well, the key piece here is we know that the body has a great defense, which is this calsequestrin. That calsequestrin, that sponge, is there to soak up that extra calcium. So if a normal amount is good, then more can be hopefully better. And that's really the prospect of this medicine, is to increase the level of calsequestrin in the sarcoplasmic reticulum to act as an extra reservoir, so that in times of either emotional stimulus or physical stimulus, with that adrenergic load, even though there's more calcium, guess what? There's more calsequestrin there to hold on to that calcium, so that we don't have that abnormal ryanodine receptor opening. So, and that's a whole lot of complicated biology, which is really, I think, and hopefully I explained it clearly and accurately, but that really is a summation of a lot of work over the last 2 decades, and a lot of it actually coming out from our collaborator, Dr. Priori, and I'll give a shout out to her in a minute. But why don't we move to the next slide, and we can talk about the components of solids gene therapy for CPVT. So this is a bit of a review, but the first thing we have is the delivery truck. This is the AAV capsid. This is the shell. This is that shell that directs to the cardiac muscle cell, and then gets into the cardiac muscle cell through a normal receptor process, and then presents genetic material to the nucleus. Then we have this on switch, the promoter. And the promoter in this case is what's called the Desmond promoter. So it promotes what? It promotes the transcription and then subsequent translation of the transgene. And this transgene is Cas-Q2 or calsequestrin, which of course we just talked about is that extra protein now on top of the normal protein that's being produced that helps to soak up that extra calcium when a patient is under any kind of stimulus, like an emotional or physical stimulus. So if we go to the next slide, as promised, my shout out to the Sylvia Priori lab. Here, this is a picture of them in Mount Jerry, where they have done a lot of this work. And really, it's really through these types of collaborations and the enduring dedication to discovery and using those discoveries to make hopefully new and better medicines that we're here. So in the next slide, what is the process that Solid undertook, to bring this new therapy into clinical trials? And that new therapy is SGT-501. We took learnings from the Dr. Priori lab. We put together a new virus, which is called SGT-501. We went into many mouse studies to look at both safety, and then we also look at mice that had been genetically modified to resemble CPVT. We use those mice to evaluate what we say is the effectiveness or the efficacy of SGT-501, this calsequestrin expressing virus, in various genetic backgrounds of CPVT. And what we saw is that those were effective in decreasing the arrhythmogenic potential, even in the setting of very high levels of adrenergic stimulation, by basically giving these mice IV adrenaline at high levels. We also conducted not only safety studies in mice, we also did safety studies in non-human primates or monkeys to make sure that we were looking at any potential safety signal that would cause us to think that this drug was not something we would want to go ahead and start clinical testing. I'm very pleased to share with you that after a thorough review of the safety and efficacy of SGT-501, we made the decision to put all that data together and to bring that to the regulators in the US and the regulators in Canada, so the FDA and Health Canada, and present this data that we said we believe the next step is to go ahead and start a clinical trial, and that's what they agreed to do. So what is ARTEMIS. So ARTEMIS is the study to evaluate SGT-501. So I'll quickly go through a study overview, which will go through who are the potential patients, participants, the potential patients that can be participants with us, and what that means to be a participant for me is really a partner. These trials cannot be done without participants, without partners, and these really, I think, are the key scientists here. I want to share with you what are those study events, what are those things that the patient does, or that patient journey as a participant in the ARTEMIS study. And then I'll share with you a little bit about what would be required from those that do decide to be our partners as a participant in ARTEMIS. So now let's talk about what ARTEMIS is. It's a phase 1b trial that's focused on the safety and tolerability of SGT-501 with patients with CPVT. And the phase 1b means that it's the first study on man, and it's being done in patients, and that's that B. The study will begin with patients who are adults, who are at least 18 years of age, and only after we have been able to evaluate carefully in adults the effects of SGT-501, we may start to open the study up to those that are less than 18, but 7 or older. This is a one-time dose, as we kind of talked about before, because it's a gene therapy that's given IV, intravenous. All patients will be receiving the study drug. So sometimes we talk about what's called a double-blind, randomized, placebo-controlled trial. There is no placebo here. All of the patients are getting the drug, and so that we can make sure that we understand its safety completely. Patients will be again followed for 5 years because this is a gene therapy. So who may participate? Initially, we're really engaging with those patients who are 18 years or older, and then it's just followed by patients that are pediatric, 7 to less than 18. Patients may or may not have an implantable cardioverter defibrillator, or ICD. Patients must have a diagnosis of CPVT, seem obvious. And then we also must require all of those patients to have a genetic test. Even if you've had a genetic test before and you have records, it's great. We'll get another genetic test just to make sure, and then we'll be evaluating that genetic test for a pathogenic or likely pathogenic in the RYR2 gene. And so there's a little verbiage there. Pathogenic or likely pathogenic is just a way that we categorize our understanding of that particular mutation. And so this would be a recognized mutation that we believe is causal for CPVT. That's what pathogenic, likely pathogenic means. Initially, all patients will have to be already on standard of care therapy for CPVT, and what that's defined as in this study is on a beta blocker and or flecainide. Patients must have had a history of ventricular arrhythmia, and we can talk about exactly what that is, but it usually would be a symptomatic event, or that they've had an event captured on a treadmill test that was clear. Because we're using an adeno-associated virus to deliver the gene, we must ensure that patients don't have antibodies to that specific adeno-associated virus. And so we will test to make sure that those potential participants wouldn't, frankly, be immune to the therapy. And then one thing that it's critically important about this study is to look for our patients still at risk for having abnormal arrhythmia after the gene therapy. And to do that, we will ask all patients to do a treadmill test during the study. And so we have to require that all patients take a treadmill, be able to do a treadmill stress test. So what does it look like if you're a patient that is decided to partner with us to be a participant? The first thing that happens is that you go to a study center, or that particular patient goes to a study center and goes through what we call a screening period where our investigator, our doctor partners at the hospital, would be evaluating all of the characteristics of that particular patient and making sure that they meet all inclusion criteria, meaning all the things that we need to make sure are included, and do not meet any exclusion criteria, meaning there's no things that would say that patient shouldn't participate in the study, for instance, if they've already had a gene therapy or something like that. Once that patient has met all inclusion and has not met any of the exclusion criteria, they are then brought to the hospital and they go for an inpatient IV infusion and a very brief hospital stay. From that day 1 of that infusion to day 30, there's a number of times that they'll be coming back into the hospital where they'll be having some blood draws to make sure that everything is going okay, and that they're being followed clinically, again, just to make sure that they're safe. Then after that we define that first kind of 30-day period as a period where there's relatively more inpatient visits, but again, they can be just local to the hospital. They don't need to be in the hospital. They then go for less frequent visits between day 30 and day 90, again, where there'll be some blood draws and also some evaluations clinically. It's really at that day 90 visit that we think about, okay, what is going on with that patient's arrhythmogenic potential? What is their potential to have an arrhythmia? And it's at that time that they have an assessment. And what that assessment is a treadmill stress test. That then is repeated at day 180, day 270, and day 360. So that assessment, being an exercise treadmill stress test, will occur at those times. And that color going from that dark blue to that lighter blue is really a way of depicting, we hope, that there's less and less dense frequent visits during that time period. And then from year 2 to year 5, we do check in with patients at roughly every 6 months to make sure that they are still doing well. We want to check in on their stress tests, and we want to make sure that their heart is functioning well. So in terms of the next slide, I believe actually this may be a duplicate, so thank you. So then what will it mean to participate in the ARTEMIS study? If you are a participant, you will be with us. We'll be partners for at least 5 years. Again, in that first 30 days, very closely, in that first year, less and less often will we be, have that kind of contact, and then really through the last 5 years, it would be a fairly infrequent follow-up. It would require around the dosing a couple of days in the hospital, and then after, in total, there's about 24 visits that patient would be part of. Again, what we're hoping to share with the density of that blue and that kind of Candyland slide is that initially the visits are quite dense, but after the first year, the visits will be far less frequent. As a partner with you, we would be supporting all of the travel, accommodations, or any inconvenience that we can. We certainly would be supporting all of our participants to make sure that this would be impacting their life as little as possible, because we understand how much it means to be a participant in a study like this. So what would you expect during a visit? One thing that we would do is we would be drawing some blood. Why would we be doing this? Well, one of the things we're looking for are changes to your immune system because of the virus, and we want to make sure that everything is going well in terms of how that response is going. And then the other things that we would be looking at is measuring your heart function. So we would take a look at that with echocardiography. And then also of course, to look at your arrhythmia. And to do that, we would be doing this exercise treadmill stress test, as well as patches to make sure that we can capture in between the stress tests any arrhythmia that might be happening in just daily living. So in summary, the ARTEMIS study is a 5-year study with most of the visits happening in that first year. The study would be dosed with a drug in the hospital with a couple of days there. Otherwise, the study would be conducted at the study center where safety checks are done and exercise stress tests are done as well. What is the study studying? It is the safety of SGT-501, which would be a single IV administration. Where is this study occurring? It's occurring in Canada, and that's why we're able to share this with you, as well as the United States. And so probably the most important slide is the one next, which is if you are interested in the ARTEMIS study, there is a QR code. You can certainly scan it, and this can give you some more details, and we do keep this up to date. So if you're watching this as a recording, it should still be available to you. Please do scan this code. Or if you just want to reach out directly, you can also use the link provided here. So with that, I would like to thank SADS Canada for inviting us today to speak with you about the basic precepts of gene therapy, the exciting news about the advancements and understanding around CPVT, that all of those advancements in that understanding has generated the prospect of what we hope is going to be an exciting new therapy for CPVT, which is called SGT-501. And then finally, how we have constructed a clinical trial and are seeking partners to become participants in that clinical trial to look at the safety of SGT-501 for ultimately the treatment of CPVT. And with that, I'll turn it back over to our hosts.
Amazing. Thank you so much, Dr. Brooks and Seth, especially for joining us today and for sharing this important information with our community. Your work and your time are truly appreciated, and we're grateful for the clarity and insight that you brought to this session. A reminder to everyone that today's webinar will be uploaded to the Canadian SADS Foundation YouTube channel. It will remain available as a resource for you to revisit or share with others at any time.